Thank you. Yes, I'll be discussing some results we've gotten from analyzing an archived set of samples that Stanley produced in 1958, where he used cyanamide, which is a plausible prebiotic condensing agent, to try to evaluate the polymerization of amino acids into larger molecules like peptides. But before I do that, I'll provide some context for this experiment. So many of you are probably aware of the original Miller-Urey experiment that he published on in 1953, where he used an apparatus like you see here on the left. He put water in this small flask, and he put the gases methane, ammonia, and hydrogen in this large flask. Over the course of 7 days, he sparked the experiment, collected the samples, and analyzed them with what was considered to be relatively state-of-the-art technology in 1953, paper chromatography with hydrogen detection. And what that did is it produced these various spots here that all represent different compounds. So in the case of his analyses, they represented various amino acids. So what this shows us is that you can simulate a possible early Earth condition using very simple starting materials, adding energy, and you can form amino acids, the building blocks for life. But recently, there's been some rejuvenated interest in some of the work that he did in the 1950s, particularly with respect to a couple different experiments, one of which was his volcanic experiment, and that was one where he used an apparatus similar to the one you saw on the previous slide, except it used an aspirator in it. And what that did was it injected a jet of steam directly into the spark discharge, simulating a volcanic eruption in the presence of lightning-rich air on the early Earth. It's something that's common today and probably was common about 3.8 billion years ago as well. And what you can see from the analyses of these samples that he produced is that you get a great range and abundance of compounds that are produced by this experiment. Interestingly enough, only a handful of these were actually detected by him when he first analyzed these samples himself, but over 5 times as many can be detected today. That speaks to the analytical achievements of the past 60 years or so. Additionally, there was his hydrogen sulfide experiment, where he used a version of the classic apparatus, and he used a reducing and oxidized gas mixture that included hydrogen sulfide, and he sparked it for about 3 days. Let's go back. Sparked it for about 3 days, and after he collected the samples, he stored them in sealed, sterilized vials, but he never looked at them. So this is the first analyses of these particular samples, and you can see a chromatogram here on the top of the samples, a chromatogram of the standards below that, and even farther below that is this little line that's an analysis of a procedural blank. So it shows you that the sample handling processes contributed negligibly, to the presence of these amino acids that were detected in this sample set. So it shows you that even by adding a simple gas like hydrogen sulfide, you can have a huge effect on the range of products, which included a number of sulfur-bearing organic molecules like methionine, which is important for eukaryotic protein synthesis. But despite these various results that have been produced from these experiments, detractors of the experiment would say, you can take simulated early Earth conditions and form amino acids, but that doesn't mean that you formed life. And that's absolutely true. And in fact, that gets at one of the biggest questions remaining in the field, which is how do you go from these small monomer molecules like amino acids into these bigger molecules like di, tri, and polypeptides, eventually working your way up into small proteins. And one possible way to do that is through the use of condensing agents. So these are simple molecules that are capable of facilitating polymerization of amino acids onto one another to form peptides. And ideally what you could do is you could demonstrate an early Earth condition, and you would introduce a condensing agent, and you would try to see if you can form both amino acids and dipeptides, which would be their immediate polymerization product, and show that, in fact, this process can happen under simulated early Earth conditions. So to give an overview of what would be a condensing agent, there are a number of possible condensing agents that could have been readily formed on the early Earth, one of which would have been cyanamide. Also, its dimer, dicyandiamide. And then cyanogen and carbonyl sulfide are also very plausible prebiotic condensing agents. But for the purposes of this study, we're going to focus on cyanamide. And that leads us to his most recent set of archive samples that have been discovered and subsequently analyzed, and that's his cyanamide spark discharge experiment. So if you look here on the left, you can see a photocopy of his original laboratory notebooks. He was, fortunately for us, a very diligent note taker. And from this, we can tell how he did the experiment. We know that he took a version of the original apparatus and sparked the gases methane, ammonia, and water over the course of 7 days. And intermittently throughout the experiment, he stopped the experiment 3 separate times to add cyanamide. And it's worth noting that after the first addition of cyanamide, he no longer heated the apparatus. And the reason for that is likely because of the risk of thermally decomposing the cyanamide. If you thermally decompose your condensing agent, you don't have a chemical mechanism or means by which to induce the polymerization of the amino acids you form. So one thing we wanted to do is we wanted to look for not only the amino acids, but also their immediate polymerization products, dipeptides, and diketopapyrazines, or DKPs, which are the cyclic dipeptide. And the reason why we want to look for those is because under equilibrium conditions, all 3 of those would be present in a polymerization reaction. So we took a targeted approach to look for amino acids, and then a separate targeted approach to look for dipeptides and DKPs. So when we looked for amino acids, we used high-performance liquid chromatography with fluorescence detection and triple quadrupole mass spectrometry. So the way this worked is we took our samples and we derivatized them. So we prepared them for analysis with a chiral adduct known as O-thaldialdehyde and acetylcysteine, or OPA, NAC for short. And what this does is it tags primary amino groups and provides an enantiomeric separation of amino acids with chiral centers. So we take our samples and we push them through an HPLC column. As the compounds of interest get eluted off of the column, they are either directed into the fluorescence detector for detection by one mechanism, and they're also directed into the triple quad for mass analysis. And if we look at our amino acid data, we can see very clearly that the cyanamide spark discharge experiment does produce amino acids, and it produced them in good yields. And in fact, it compares very well to the previous spark discharge experiments that he had conducted. What's worth noting is that you have the presence here of the aminobutyric acids, which are non-protein amino acids, and you also have amino acids that are present in high abundance that have chiral centers. They are racemic within experimental measures. So that suggests to us, the combination of the 2 factors, suggests that these amino acids were formed within the experiment itself and were not a product of sample handling processes or other sources of terrestrial contamination. Now, if we look at dipeptides and DKPs, our approach for looking at these was to use ultra-high performance liquid chromatography with quadrupole ion mobility separation and time of flight mass spectrometry. So when your compounds of interest come off the column, they come into the instrument here and they get ionized. And that's when they go through ion mobility separation, which separates the molecules based on their cross-sectional area when it interacts a drift gas acting in the opposite direction. And then they enter into the mass spectrometer here for high-resolution mass analysis. And when we look at our dipeptide results, we do see a lot of dipeptides, and we see them in good yields as well. And interestingly, if you look at your amino acid to dipeptide ratio, the ratio in the samples is about 1,000 to 1 or 1,000 to 10, which agrees with experimental evidence that shows that at equilibrium conditions, you should have amino acids in about 1,000 times more abundant than your dipeptide. So that suggests that these dipeptides were formed within the experiment and were not a product of contamination. If you look at the structures down here at the bottom, that shows you the type of chemical complexity that we're dealing with, the kind of complexity that we're looking for. We're not looking for super large macromolecules. We're looking for something a little bit larger than amino acids. So let's look at our DKPs. We do find DKPs as well. And this is very good evidence because it shows the full product of the polymerization of the amino acids. So you see DKPs in high abundance, and when you compare your dipeptide to DKP abundance, it's about 1 to 10 or 1 to 20, which generally agrees with experimental evidence that shows that at equilibrium conditions, your dipeptide to DKP ratio should be about 1 to 10, further suggesting that these DKPs and these dipeptides were not a product of contamination. So one question would remain from this type of study, which is, how does this happen? What's the mechanism by which this might occur? So to take a step in a direction of trying to address this question, we're proposing a mechanism. And it starts like this. You have cyanamide, and at equilibrium, cyanamide would also be present as carbodiamide. This carbodiamide can be protonated, and then when protonated, it can react with an amino acid, like glycine in this case, which would form the activated form of the amino acid. So here would be activated glycine. The activated form of the amino acid can then react with another amino acid to yield your dipeptide. Your dipeptide can then form your DKP through two different means, one of which would be a dehydration reaction, the other one of which would be direct reaction with cyanamide itself to yield your DKP. So from this study, there's several things we can conclude. First thing would be that this was chronologically the first attempt to study condensing agents for their relevance to the origins of life. Additionally, we can conclude is that, yes, you can form simultaneously both amino acids and their polymerization products, dipeptides and DKPs, under plausible simulated early Earth conditions, and that these condensing agents, like cyanamide, would provide us a chemical mechanism by which we can explain how this polymerization may have taken place on the early Earth, and this could have expanded upon and diversified the prebiotic chemical inventory from which life may have originated. So with that, I should acknowledge colleagues, collaborators, funding, and this resource down here, which provided us with access to Stanley's lab notebooks, so we can understand how he did the work he did. Thank you.
All right, thank you very much. Do we have any questions? All right, Guillaume.
Hi, thanks for your talk. I'm just wondering, on these types of experiment right now, what's the level of complexity we can achieve? I know you were looking for a specific level of complexity, but how long peptide chains can you synthesize abiotically in these type of experiment?
It's a good question. So the question was, what's the type of complexity that we can reasonably expect from these types of experiments? And a lot of it depends on the variables that you introduce into the experiment. What are your starting conditions? How long do you let the experiment go on for? So he would let the experiment run on average for a week. If you let it run for probably 2 or 3 weeks, you can generate even greater amounts of complexity. So, and it also depends on subsequent compounds or components that you may introduce into the reaction apparatus. So in this case, he put a condensing agent. If he continued to add more condensing agents, maybe a greater variety of condensing agents, that could have facilitated polymerization into polypeptides. So one thing I didn't mention in this talk was we focused on dipeptides, but we also did screen for tri and polypeptides, and we did see tripeptides and very small polypeptides, but we didn't quantitate that. So you can reasonably expect polypeptides, I'm confident in saying at this point, and we're also in the process of looking for things like nucleobases and moving on beyond just the simple monomer amino acids.
All right, do we have one more? Okay, thanks very much, Eric. Yep, I lied.
Thanks for a great talk, Eric. I don't know much about condensing agents. Are cyanamide or other condensing agents things that you find in the natural environment in abundances comparable to those that that Stanley Miller actually used?
Say that again?
Are condensing agents like the ones that were used in this experiment, or other condensing agents, are those things that are commonly found in the environment and in concentrations comparable to those used here?
Yes, they are. So these condensing agents that I discussed are readily formed under simulated early Earth conditions. So they're very simple molecules. They're formed from the starting materials that were likely present on the early Earth and using the energy sources that were likely present on the early Earth, like UV radiation. So for example, cyanamide is formed from the ultraviolet radiation of ammonium cyanide, which is formed fairly readily on the early Earth. So these are very much plausible molecules, and the concentrations he used in this experiment were on the order of millimolar to micromolar ranges, which is appropriate for an early Earth setting.
Quick follow-up to that, is cyanamide ever found in the experiments where condensing agents weren't added?
It's a good question. So we're looking for cyanamide in spark discharge experiments. There's currently no literature that shows that cyanamide has been formed in spark discharge experiments. One problem with that is a lot of the analytical instrumentation is mass spectrometry based. So if you're looking for a really low mass molecule in the aqueous phase, It's going to be tough to find it using mass spectrometry because there's certain mass cutoffs. A lot of mass specs will not have very good accuracy or precision for masses below 50. So cyanamide is around 42 is their AMU. So it's difficult to look for that. But you can use other things. You can use chromatographic techniques. But that's something that we're currently looking for, is to see if it can be formed inherently within the spark discharge experiment.
All right, thanks very much, Eric.